Today's topic is about the regulation of glycogenolysis by hormones, main negative regulator of glycogenolysis, the insulin. First of all, let's wrap up the idea that glycogenolysis is the pathway that's responsible for breaking down the glycogen, sugar stores present inside the liver, into glucose-free inside the bloodstream. However, the glycogenesis is the pathway that stores the glucose present in the blood in the form of glycogen inside the liver. Amazing. How insulin will regulate the glycogenolysis? Okay, insulin is an enzyme that's secreted by beta pancreatic cells of the pancreas for the aim of storing excess glucose sugar present in bloodstream inside the liver in the form of glycogen. First of all, let's start and talk about the receptor of insulin. In the previous videos, when we talked about the regulation of glycogenolysis through epinephrine and the glucagon and epinephrine binds on alpha and beta receptors, a G protein coupled kind of receptors. However, the insulin receptor that is present on the surface of hepatocytes is a tyrosine receptor and not a G protein coupled receptor. Alright, now the insulin, when glucose concentration exceeds the limit of normal blood glucose concentration, insulin will be released, bind on its receptors that will send signals responsible for the shutdown of glycogenolysis. So glycogenolysis, as we said, it's the pathway that breaks down sugar stores in the liver known as glycogen into free glucose inside the bloodstream. So this insulin works on shutting down this pathway, and when we shut down the glycogenolysis, automatically glycogenesis, the opposite pathway, is responsible for taking excess glucose present in the blood into storing it inside the liver. So this insulin will work on shutting down the glycogenolysis and activating glycogenesis by taking the excess glucose present inside the blood and store it. How this will occur? First of all, in order for insulin to perform such signaling, the receptor, because it's a tyrosine kind of receptor, it should be dimerized. So the receptor should be dimerized in order to phosphorylate effector proteins. These effector proteins present inside the liver will perform a variety of roles that we will talk about in a moment. Undergo phosphorylation. Tyrosine receptors are a kind of receptor that, in order to be active, they should be dimerized, and once dimerized, there will be transphosphorylation. Two monomers linked together are called dimers. So the tyrosine receptors will be linked, two monomers, two tyrosine receptors will be attached together. This attachment is called dimerization. Once they are dimerized, they will undergo transphosphorylation, which means one monomer will phosphorylate the opposite monomer attached to it by this activating the receptor. Once the receptor is active, the signal will be transmitted inside the cell and activate the glycogenesis. Main role of effector proteins, AntiGlucagon Effects. Glucagon is a hormone also released from the pancreas, works on activating glycogenolysis, which means the breakdown of sugar stores inside the liver into free glucose inside the blood, which is opposite to the role of insulin, where insulin, as we said, is released only when the glucose levels increases in the blood, and we work on storing it inside the liver. However, the glucagon is at the opposite. How the anti-glucagon effects will occur? First of all, we will increase the GTPase. As we mentioned in previous videos when we talked about the effect of glucagon or how glucagon activates glycogenolysis, there is a glucagon binding receptors. The beta receptors are G protein coupled receptors, which means a receptor coupled with alpha, beta, delta subunits and with a GDP molecule. Once the receptor is coupled with a GDP, the receptor is inactive, which means there is no transmission of signals. Once this GDP molecule is transferred into a GTP, the receptor will be active and the glucagon will be able to transmit its signal and activate glycogenolysis. The role of effector proteins is to increase GTPase, which means an enzyme that will remove the phosphate group from a GTP, converting it back into GDP, the inactive form of glucagon receptors. And once this will happen, the glucagon receptor will be always inactive, which means no signaling of glucagon, no activation of glycogenolysis. And once glycogenolysis isn't active, glycogenesis automatically will be active. Pathway that insulin need to be on in order to store the glucose present inside the blood, inside the liver in the form of glycogen. Second role of effector proteins is the increase of phosphodiesterase. As we mentioned in the video of regulation of glycogenolysis through epinephrine and the glucagon, when epinephrine binds on alpha receptors, it acts different than that of role it acts on beta receptors. Where on alpha receptors, epinephrine will lead to the presence of a molecule called isotyl triphosphate. This isotyl triphosphate will be responsible for the activation of calcium channels that will lead to the formation of a complex called calcium calmodulin dependent protein kinase that's responsible for the activation of glycogenolysis. Let's not go deep into details. Where this phosphodiesterase will work on removing a phosphatic group from this isotyl triphosphate IP3, leading to its inactive form isotyl diphosphate IP2. This IP2 won't perform the role of activating the calcium channels. The calcium calmodulin complex won't be present. No activation of glycogenolysis, and by this we will inactivate the glycogenolysis through a second pathway than that role performed by GTPase. Also, there is the phosphoprotein phosphatase. As we mentioned, the role of glucagon and epinephrine on beta receptors, they lead to the activation of protein kinase A. This protein kinase A main role is to activate glycogenolysis, which means the release of glucose from its stores inside the liver outside into the blood as a free glucose molecule. How this will occur? It's all about phosphorylations performed by this protein kinase A. These phosphorylations will lead to the inactivation of glycogenesis, the pathway that stores glucose from blood into the liver, also by activating a variety of enzymes. However, when phosphoprotein phosphatase is present and active through insulin, these phosphorylations performed by protein kinase A for the activation of glycogenolysis won't be present, which means no phosphorylation of proteins. When there is no phosphorylations by protein kinase A, no activation of glycogenolysis, and hence no release of free glucose molecules inside the blood. And once the glycogenolysis isn't active, automatically glycogenesis, the opposite pathway, will be active, which is the main aim of insulin, and by this the glucose present in excess inside the blood will be stored inside the liver. Finally, insulin will perform the increase of GLUT4 receptors. What are GLUT4 receptors? On the surface of muscles there will be receptors called the GLUT4. This GLUT4 will take glucose present in the bloodstream and allow its entrance inside the muscle cells that then will be recruited into many pathways in order to be stored. Once this GLUT4 expression increases, the glucose will be accelerated, and by this we will decrease the concentration of glucose from the blood, reaching the normal limits we are seeking for. By this we will end the topic of today. Hope I make it clear for you. Don't forget to subscribe, and I will leave links in the description for other YouTube videos. Thanks a lot.